Welcome to AP Biofunnel with Dr. D, and in this tutorial we continue with phylogeny or phylogenetics, which is part of Unit 7. And now we're going to talk a little bit about cladograms. So cladograms are a type, cladograms are a type of phylogenetic trees. Cladogram is used to show evolutionary relationship amongst different groups based on traits. So cladograms usually have markings on them which show traits. These traits are called shared derived characteristics. Derived characteristic is a trait that has evolved only within a specific group under study, for instance, feathers and birds. A cladogram has clades. A clade is a grouping that includes a common ancestor and all of the descendants, both living and extinct, of that ancestor. Using phylogeny, it is easy to tell if a group of lineages forms a clade. Imagine a clipping of a single branch of the phylogeny. All of the organisms on that pruned branch make up a clade. So what's in a clade? Each colored rectangle below represents a clade. And what I want you to notice is that the clade will contain the branch point, which is the common ancestor, as well as the two branches. So the two descendants, if you look here, and the yellow clade. A clade can also be nested within a larger clade. So here's a common ancestor, one of its descendants right here. And then the second descendant of that ancestor is the common ancestor of these two, right? So the yellow clade, the yellow group, is nested within the larger blue group, and the blue group is nested within the larger green group. Again, the ancestor and all of their ancestors, descendants. So here are some examples of a clade, not a clade. A is not a clade right here because it's only one descendant and not an ancestor. Here we have a common ancestor, two descendants, but a descendant from a different common ancestor. So it's also not a clade. This one is also not a clade because it is missing one of the descendants. If that orange were to include the last part here, it would be considered a clade. Okay, so clades again are nested within one another and they form a nested hierarchy. A clade may include many thousands of species or just a few. A clade may include species that are no longer living and they're extinct. So in comparison with its ancestor, an organism has both a shared and different characteristics, right? Remember, descent with modification. So we share characteristics with our common ancestor, but also different. A shared ancestral character is a character that has originated in the ancestor of the taxon. A shared derived character is an evolutionary novelty which is unique to a particular clade. And a character can be both ancestral and derived depending on the context. And here's my favorite cladogram. This cladogram shows the shared derived characteristics of vertebrate animals. Vertebrate animals have a vertebrate column, okay? So you can see right here at the bottom is the shared characteristics of all the clade, so this characteristic is a characteristic of the big clade, but it's also a characteristic that is shared by all the nested clades, that is vertebrae, okay? Then you will see a derived characteristic, which is bony skeleton. So sharks do not have bony skeletons, but all the rest of them have. Then the next trait that was developed were four limbs, hair, two orbital fenestra, and so on. Here is the most important shared derived characteristic of land animals, amniotic egg. Amniotic egg is the ability of the developing embryo to be surrounded by fluid, the amniotic fluid, which enabled animals to colonize land. So they didn't need water to lay their eggs, to provide a source of water for the developing embryo, because their embryos were enclosed in an amniotic sac, which had its own fluid in it. So when trees are constructed, the principle of maximum parsimony is at play. Maximum parsimony assumes that the tree that requires the fewest evolutionary events is the most likely. And parsimony is a word that I didn't know what it meant until I looked it up years ago. It means being frugal. So parsimony is based on that first principle that we talked about last lecture, that the closer related two species are, the fewer changes there are between them, or the more similar they are, right? The more similar they are, that means the fewer changes there are. So conversely, we assume that evolution, which proceeds slowly, is very parsimonious, it's very frugal, it's very stingy. So the fewer changes it requires for a species to evolve, the more closely. So when we're grouping species, we're grouping them based on the two species that will be most closely related on the tree, placed on the tree will be the ones with fewer changes, with fewer differences, because it assumes that the fewest changes are the most likely explanation for how the species have evolved. And this can be based both on differences in DNA sequences as well as differences in presence or absence of character. So in this case, we have a phylogenetic tree based on molecular data. There are three possible hypotheses to explain how these three species, which are very closely related, have evolved. And based on molecular data, which tells us how many events were required for each of these models, it turns out that only one of these models is correct because it requires the fewest number of changes and events. You can apply the same concept to the presence or absence of characters. And by the way, you will probably be asked on the exam to construct cladograms or construct phylogenetic trees based either on characters, certain morphological characteristics, or based on DNA sequences. And what you will always do is you will start with the outlier. In this case, the outlier or the outgroup is the landslide, because the landslide does not share any of the traits the others do. And then the more traits two organisms share, the more closely related they are, okay? So based on all these traits, you can group the organism, you can build a model of how the organisms are related. Now we can also add time to a tree, and this tree here shows you time. Not every tree is going to have a scale that shows millions of years, but in general, the length of a branch corresponds to length in time. Shorter branches, so for instance, you can see here that birds, this is the length of branch for the birds, but then in their clade, there is a species where the branch is much shorter, and that means that's an extinct lineage. So how do we add time to a tree? Well, the length of branches does reflect time, but in the first case that I'm showing you here, the length of the branch reflects the number of genetic changes that have taken place in a particular DNA sequence. But that relies on the following concept. The more changes that have accumulated in a DNA, the more time has passed, evolutionary time, because changes in our DNA, mutations in our DNA occur at a predictable rate. So the more changes, the more time has passed. So again, this is an indirect way to show time, but in this case, they're based on specifically on number of genetic differences. In other trees, the length of the branch reflects directly time, and that time can be determined either from the fossil record or molecular clocks. So in this case, here we have the tree, and in this case, we have a scale which shows us millions of years since the last common ancestor humans and mouse had were 65.5 millions of years. Okay, the last common ancestor humans and chickens had was right here, somewhere like 240 something millions of years. Okay, so what is a molecular clock? Molecular clocks help us track evolutionary time. They extend molecular phylogenies beyond the fossil record. So the fossil record helps us with time, but molecular clocks can further extend that. And in order to do that, we must make an assumption about how molecular change occurs over time. Evolutionary changes may actually occur in a clock-like fashion. Since changes in DNA sequence can build up over the course of millions of years at a reliable rate. So there's certain regions in the genomes of organisms where we actually know what the rate at which changes accumulate is. And that rate is predictable, and we can use those sequences as a molecular clock. So different organisms have different rates of mutations, and also different DNA sequences can have different rates of mutations. And there's certain places in the genomes of organisms where those changes occur at a predictable rate, and these places are used as molecular clocks. And if you remember, I showed you a phylogenetic tree for the globin genes that build hemoglobin, myoglobin, and fetal hemoglobin. The alpha globin gene is one such molecular clock. It experiences base changes at a rate of 0.56 changes per base pair per billion of years. So by determining the differences in alpha globin genes between organisms, we can determine when was the last time they had a common ancestor, because we can calculate, so how many changes are there? If there's one change, right, then we can say that the last common ancestor we had with that organism was approximately 2 billion years ago. So this is how molecular clocks work, and they're quite reliable. So we can also apply molecular clocks to the dating to analyzing human diseases. And I'm going to give you one example here, but I'm also going to talk a little bit about another example. So viruses are not considered living, but they are subject to evolution. They constantly change and mutate. They adapt to evade our immune system. They adapt to evade any type of immune system to a host or in a host organism, not just humans. They adapt to medications, to any type of treatment. And HIV, the human immunodeficiency virus, has been around for quite a long time. And molecular phylogenetic analysis shows that it is descendant from viruses that were infecting chimpanzees and other primates. So it is the original virus, before it started mutating, was a virus that infected primates. Now that virus jumped to humans, and it turns out that HIV spread to humans more than once in the history of life. And if you compare all the different HIV strains, all the different HIV samples, it shows that the viruses actually evolved in a clockwise fashion. And surprisingly, it turns out that one strain of the HIV virus has spread to humans as early as the 1930s. So most recently, a molecular phylogeny has been done and continues to be done on SARS-CoV-2, or the virus that causes COVID-19. And the difference, there's quite a few strains now. And these strains are compared to other coronaviruses, to SARS-1. And that molecular analysis actually definitively has been able to disprove the claim that the virus was generated in the lab, for instance. And it can actually be quite useful to trace the spread of the different strains and the evolution of those strains. So it helps us really tackle human diseases. So as I said, identifying the source of new human diseases is one way phylogeny is useful. So in addition to using phylogenies just to classify organisms for taxonomy, it is very useful to identify the source of new human diseases. It is being used to set conservation priorities. It's used in agriculture. It is also used to select model organisms in biomedical science. So if you would like to, for instance, study how cell cycle is regulated, because unregulated cell cycle can lead to cancer, and whether certain drugs can actually help treat cancer patients because they interfere with cell cycle progression. You're going to choose certain organisms to study this in, because we cannot study directly in humans, and you're going to choose those organisms based on evolutionary relatedness to humans, and they're called model organisms because we model human diseases and treatment to human diseases in these organisms. Okay, I would like to also point out a few things which actually relate to our last topic, which is the origin of life on Earth, is that here is a very broad bird's eye view of the tree of life as we understand it currently. The common ancestor to all life is shown in blue, and then we have now grouped life into 3 domains: domain bacteria, domain archaea, and domain eukarya. Domain bacteria contains all the common prokaryotes, all the common bacteria that we know, everyday bacteria. Domain archaea contains some ancient microorganism or ancient bacteria. Archaea comes from the Greek word archaic, old, ancient. Domain eukarya is all the eukaryotic organisms. And I want you to notice one thing here. Everything on this tree, except for the 3 kingdoms shown in blue, which is land plants, fungi, and animals, is single-celled organism. So the majority of life on Earth is simple still, believe it or not. Single-celled organisms. Only animals, fungi, and land plants are actually multicellular. Now, I want to talk a little bit about horizontal gene transfer. I'm going to remind you that horizontal gene transfer is, let me just go right here, the movement of genes from one genome to another, not during so a vertical gene transfer is what happens during sexual reproduction. Horizontal gene transfer happens without sexual reproduction. It's the movement of genes from one organism to another, from one genome to another. And we talked a lot about horizontal gene transfer in bacteria, but it does happen elsewhere as well, or at least we have evidence that it has happened. So remember, in bacteria, it occurs by exchange of transposable elements, those jumping genes, by plasmids, by viral infection. It can also happen via fusion of organisms. And we have those jumping genes, we have those transposons, and they're believed to be remnants of viruses. And there are evidence for horizontal gene transfer. Now, horizontal gene transfer complicates our efforts to build the tree of life, and I'll explain in a second why that is. Now, we believe that there has been a substantial interchanges of genes between organisms in different domains. So if you look at the tree of life right here, we know for certain that archaea, domain archaea, and domain eukarya had a more recent common ancestor than the common ancestor of all life, which means they're more closely related to each other than they are to bacteria. So if we apply the same principles that we did in the first lecture, common ancestor, speciation event, right? Although this is more of a larger scale change. One result is the evolution of domain bacteria. Another result would be the common ancestor of both eukarya and archaea, which means archaea and eukarya, eukaryotes and ancient bacteria are more closely related to each other than they are to bacteria. So how do we know this? We know this by analyzing the genes that code ribosomal RNAs. Genes that code ribosomal RNAs, RNA genes, so the ribosomal RNA genes, evolve very, very slowly, and they allow the detection of homologies between distantly related organisms. So these genes indicate different evolutionary relationships than what you normally would think if you were to just look at the basic characteristics of these 3 domains. So if you consider, however, gene transfer, that may explain some of what we observe with the ribosomal RNA genes. And if you consider horizontal gene transfer, if we can consider that horizontal gene transfer actually happened quite frequently in the early history of life, when life was very simple, single-celled, then instead of drawing a perfectly branched trees, What would be more accurate is to depict the tree of life as a tangled web rather than a branched trees. And I'm sure you're going to recognize these two tangles right here, mitochondria and chloroplast, the results of endosymbiosis. There are essentially fusions between domain bacteria and domain eukarya. So we do actually have evidence for horizontal gene transfer, because we know domain bacteria and domain eukarya mixed. How do they mixed? When domain eukarya swallowed, when an organism from domain eukarya swallowed an organism from domain bacteria and they became endosymbionts, such as in the case of mitochondria and chloroplast, that is evidence for horizontal gene transfer. Fusion of two organisms resulted in eukaryotes having now mitochondria and chloroplast. So if we're actually quite, so for these two branches, we know that we have to draw the tree this way. The other ones are just hypotheses, but the truth is that this is what the tree of life most likely looked like in ancient times if we go back to very, very billions and billions of years ago.